Thank you, Tammy. It's my great pleasure to have the opportunity to introduce today's plenary speaker, Sinead Collins. And we're very lucky to have her here. There's something very unique about her research, and that is it bridges across several subdisciplines. So I think whatever she says today will make someone or everyone actually here happy today. She will cover oceanography because she has worked with ocean acidification. She also covers limnology because she works with a model species, which is a limnic phytoplankton species. And most of all, she covers evolutionary biology, and that's really her main field. And I think it's a great thing to have the evolutionary biology and experimental evolution brought into ASLO to a much bigger extent than it has before. So I think Sinead has made a very good decision to come here today. She has also made some other good decisions in her life. So she started out as a chemist, but after a while she figured out that biologists are much more fun. So she decided to do a PhD in evolutionary biology at McGill University in Montreal. And after she finished her PhD, she made another good decision. She realized it's wonderful to live and work in Europe. And so Sinead went to Germany, to the Max Planck Institute in Cologne, where she did a postdoc. And then she continued on to Edinburgh in the UK. And now she actually has a permanent position at the University of Edinburgh. And with that, I will give the word to Sinead.
Hello. So I changed the title of my talk to make it what I really mean, which is what my entire lab is obsessed with doing, which is predicting evolution in life-giving slime. And we're not picky about our slime. We'll use freshwater or saltwater slime. We've even been known to use mud slime. And by slime, I mean single-celled algae. So the system I work most broadly in, I guess, are marine systems, where I'm really interested in understanding how primary productivity is going to change in the very productive bits of the ocean over the next decades, over the coming decades. And what's, I think, a really important bit of this question is that if you want to know how traits will change, you need to know which traits natural selection sees. You can't approach this problem simply by asking, will things evolve? I'm like, they're microbes, of course they will evolve. This is not a question. How will they evolve is the question. Another thing is, that this isn't a question that you can approach purely with metagenomics, no matter how many metagenomes you metagenomic. Until you know what traits those link to and whether or not natural selection can see those traits, metagenomics is still not at a level where it can answer this question on its own. And I'm not going to be talking any more about genetic approaches, partly because I do very little with it, and I feel like so many other people in the field do it. Yeah, that niche is full. So, predicting evolution in life-giving slime. This is sort of where we are with our knowledge of ocean acidification research and climate change studies. We study single drivers on single species, often single genotypes, and we study them over very short timescales. This is what we know. This is where most studies are. There are studies in communities. There are studies at long-term data sets, and we've heard a lot about them in this conference, which is really great. But still, the majority of studies are down here. But what we want to know is how whole ecosystems will respond to multiple drivers over timescales that can incorporate evolutionary responses. So while all of these axes on the magical cube are useful and interesting, I'm going to tackle just one monster today. I'm going to talk about how we can scale up from looking at evolution in response to single drivers to evolution in response to multiple drivers. And then I'm going to sort of play with that because there are some really interesting results that were coming out of it, and start talking about how we can predict, or if we can predict, which traits will evolve and which traits natural selection will see. And this is how I often feel beginning of experiments. This is Austriococcus in the form of an Austriokraken. In my lab, we feel like people respond better to model organisms if they think it's charismatic macrofauna. So we oblige them with artwork. So just to give you an idea, I'm sure you all know this. This is looking at ocean acidification. It's looking at climate change in salty systems. So now we have a system where CO2 flux is low relative to what it's going to be in the future. There's less deaths and fewer storms than there are going to be in the future. There's more nutrients than there will be in the future, and the photic zone is deeper now than it will be. And this is a vast oversimplification because I'm talking about sort of global averages here, and no species lives at a global average. But the point I'm trying to make with this slide is that it's not a single bit of the environment that is going to change at once. It's the whole environment in all of its glory, and then superimposed on that, there's going to be species interactions, right? So it's just going to be a general glorious mess. In addition, evolutionary biology tends to think about environments that are always getting worse. But if you're photosynthetic, CO2 enrichment can be seen as an environmental improvement, and indeed, for many plants, including a lot of algae, if you take a system, warm it up, and add CO2, they actually grow faster. From the organismal point of view, for quite a few organisms, some of these changes here are, on balance, environmental improvement. And I'm going to come back to that later, because this is something that evolutionary biologists are really uncomfortable thinking about. We're sort of pessimistic. We prefer to think about environments deteriorating. So we started out in my lab thinking, excellent, we're going to do an experiment, a nice long experiment, maybe a few hundred generations. We'll use Chlamydomonas because it's a good workhorse organism. It's a freshwater microalgae. Our Chlamydomonas has been in the lab since about 1960, which predates me. So don't think of it as a real organism. Think of it as a wet simulation. Okay, this organism probably doesn't do anything that a normal Chlamydomonas would do if you went and found it in a lake or in a field. But we thought, nonetheless, the great thing about this is we can grow it at really high densities. We know exactly what it should do. So we're going to use it for this big experiment. And there's not a lot of theory about how evolutionary response to multiple drivers should differ from evolutionary response to a single driver. However, there is a vast body of theory in evo-devo about how more complex organisms, organisms with more traits, should evolve more slowly than organisms with fewer traits. It's called the cost of complexity. And the logic goes something like this. And a lot of this has been debunked, but this is nonetheless the logic. The logic is that if you have more complex organisms, those organisms have more traits under selection. If you have more traits under selection, you have more opportunities for epistatic problems. Because of this, it also means that any given mutation is going to have a smaller effect on fitness, and you get slower evolution. This is not biology, this is geometry. If you think about an organism with two traits, X and Y, with some optimum trait value, 0, 0, and you start off at some distance from the optimum and move by novel mutations fixing, you see that each mutation will have some size. If you then have three traits here, and you take mutations of the same size, and you move them through three-dimensional space, each step is going to take you a smaller proportional distance to the optimum. There is absolutely no biology in this, okay? And that's the basis for the ideas about the cost of complexity. And we thought, excellent, this is a good place to start. We'll start here. And we made a really big assumption. Our really big assumption was that if there are more things changing about the environment, you're going to have more traits under selection. Our experiment didn't require that we know the shape of this relationship, only that the slope be positive, and that we were in the part of the relationship where the slope was positive. So as we added more and more environmental drivers to our changing environments, more and more traits would be under selection. We were, of course, wrong. However, we didn't realize that until afterwards. So this is the PhD work of Georgina Brennan. We use Chlamydomonas reinhardtii. Georgina is now doing a postdoc looking at bumblebee pheromones. I'm not sure about the link. But what we did was we looked at evolutionary responses of Chlamydomonas in response to all of these single environmental changes, as well as combinations of 2, 3, 4, 5, 6, 7, and the single 8 driver combination of these changes. Now, those of you who are statisticians are probably already worried about this experimental design, but just stick with me for 2 more slides, okay? So we used a bunch of different environments for each level of number of drivers. So for instance, we had many different 4-driver environments. So by comparing how evolution differed within the same level of environmental drivers, but between different identities, we could look at how the identity of the environmental driver affected evolution independently of how many things were changing. So here we have environments that differ in that this one has high UVB and this one has low nutrients. Once we've taken that variation into account, we could compare over levels of environmental drivers to see how the number of environmental drivers affects evolution independently of their identity. So this is just asking, this is just asking, when you make the problem more complex, how does that affect evolution? The experiment looked like this. We started with a single clone of Chlamydomonas, and the reason we do that is to set genetic variation to 0 at the beginning of the experiment so that we know that any evolution we see is novel genetic or epigenetic changes. We had a control environment. It's not important what this is. It's the environment that Chlammy has been growing in my lab since 2001. So it's the environment it's used to, all right? I took this Chlammy with me from Montreal. We had 8 different single driver environments, and then we had a bunch of different environments with 2, 3, 4, 5, 6, 7, and 8 drivers. There is, of course, only a single way to choose 8 drivers. This is the number of possible regimes. A regime is just a unique environment for each number of drivers. That would have given us 256 selection environments, and even in my lab, that's a bit much. So what we did was we picked a subset of regimes, and we ended up with 96 different selection environments. This wasn't a random subset. The experiment was also designed to pick out the genetic basis of adaptation to high CO2 with and without other drivers present. And I'm not going to talk about that today, but exactly half of these environments are high CO2 environments, and half of them are low CO2 environments. And we evolved Chlammy in these 96 different environments for 350 generations, and in each environment we had 6 replicate populations. I hope the statisticians are satisfied. I was satisfied. Georgina was, at the beginning of her PhD, she probably didn't understand what she was getting into. So the question we asked is it easier or harder? Do you adapt more or less when there are more environmental drivers present as your challenge? So is it harder to adapt to environments with more drivers? I feel like at this point a vote would be appropriate. Your vote will not influence the actual data on the next slide. So who thinks that it's harder to adapt when there are more environmental drivers? Okay, we have people voting. That's good. High voter turnout is important. Who thinks it's easier to adapt when there's more environmental drivers? Okay, that's very close. Who thinks it's not about the number of environmental drivers, but what those drivers are? It's only about the identity of the... Okay, so some people voted twice. That's okay, Roxy. Some people voted 3 times. I don't know the name of the person I saw vote 3 times, but that's called bet hedging, and I'm sure there was a talk on that at some point. So the answer is, it's not harder to adapt when there are more environmental drivers. It's in fact easier. I'm going to walk you through this slide, just in case all of you don't spend your time counting green dots under a microscope. So what we're looking here is the average cell division rate per day, the growth rate of Chlamydomonas. These things never reach carrying capacity in the experiment. So this is just sort of they just sort of turn over at a fairly steady rate. This is the number of environmental drivers. Each dot is the average of the 6 replicate populations in that regime. The gray bars, the light gray bars, are control populations grown in these regimes, so it's the first time they see these regimes. The dark bars are populations that evolved in those regimes. So the difference between the light and the dark bar is the average amount of evolution that happened at that level of environmental drivers. All right. If there were between 1 and 6 environmental drivers, we saw no populations go extinct. If there were 7 environmental drivers, we saw some population extinctions, which we just noted and then rescued. We were able to rescue all of them by artificially jacking up their population sizes. And with 8 drivers, all of the populations went extinct, even though initially they didn't have a growth rate response that was so negative that we expected them to. So this extinction was a slow and painful and gradual process that happened over about 300 generations. Okay, it wasn't something we expected at the beginning. Their growth rate actually declined over time. It didn't drop and then hold steady. So what you see is at higher levels of environmental drivers, when there are more environmental drivers, the populations that evolved in those regimes are doing better than the control, the first time the control sees it, whereas when there are few environmental drivers, the evolved populations don't do better than the controls. But there's an important caveat here. The controls are actually pretty happy here. So if I plunk you into an environment where you're pretty happy anyway, you're probably not going to adapt a lot, right? Selection is going to be quite weak in that environment. Whereas here, the controls are deeply traumatized, right? So these are environments that, for lack of better terms, suck. So adaptation is possible because you're starting from a very low point. And in fact, what we see is once we take the initial drop in fitness into account, that we don't need to know anything about the environmental drivers, that just explains, that is our main driver of how much evolution happens. So it's not harder to adapt when there are more drivers happening. All that matters is how much your fitness drops initially. I'll just go back and point out that the fitness drop for, it really doesn't like it when I go back. The fitness drop is the same for all of these sort of high levels of environmental drivers. So it seems to be once the environment is bad, you can't make it much more bad until you make it lethal. So it's another paper which I'm not going to be talking about today. So what happened? We were really surprised by this idea that no matter how much more complex we made the problem of adapting, it didn't seem to matter. If your initial drop in fitness was high, but you managed to survive it, you adapted. And it didn't matter whether that initial drop in fitness was attributable to 3 changes or 7 changes in the environment. Well, one possible explanation is that adaptation is actually driven by very few dominant drivers, so the problem doesn't get more complex. Another possibility is that the number of traits, well, a related possibility would mean that the number of traits is not going up much as the number of environmental drivers goes up. So if only a few key drivers determine what's going on, you're always going to have the same number of traits under selection as the environment gets worse and worse, but they're just going to be under stronger selection. Not too interesting. Another thing is that the levels of epistasis could be not as high as predicted by theory or as is necessary to incur a cost of complexity, and other experiments back that up. We think in reality that it was some combination of these, but I do have some evidence, at least, that the number of traits under selection is not going up as the number of environmental drivers goes up. But interestingly, I can't attribute the responses to a few dominant drivers. And the way I'm going to talk about that is by talking about local adaptation next. So I think 2 interesting things came out of this experiment, besides the fact that Georgina's a trooper. Because really, people, come on, it's like 600 cultures. And then when you do the fitness assays, it's like 600 times 6 cultures, right? It's crazy. It's completely nuts. So one question I had is, why aren't these growing faster? So in the single driver environment, some of the environments were pretty terrible, right? If you're Chlamydomonas and I take away all of your phosphate, that is not so fun. That's a terrible environment. But they didn't adapt very much. And in cases where the environment was originally enriched, so where the first thing we did was give them more carbon dioxide and their growth rate went up a little bit, they actually never got faster than that. Their growth rate went up and then just stayed up. So evolution couldn't do better. So I was interested in why evolution couldn't do better here. And I was also interested in whether or not these were generalists. So the question I'm asking with the high net environments is, does Chlamydomonas, do these organisms see each of these high NED environments as its own special snowflake environment? So if I have 2 environments, each of which has 5 drivers, and they differ only in whether or not they have UVB irradiation, does Chlamydomonas see 2 environments that are very similar? Or does it adapt to each of those environments as a completely different environment? This is really important. The reason this is really important is because so much of the way we approach trying to predict what will happen as oceans acidify is by using scenarios. So this question about local adaptation is asking how important is it that we get our scenarios right if we want to know how the organisms are going to respond. Does that make sense? Because if you can screw up the entire response or get a completely different response because you change one variable, that's a little bit iffy in terms of trying to understand how organisms respond to suites of variables. And that's not even taking into account that you have to get the levels of environmental change right, like the intensity of each variable right. So, oh yeah, there was one more thing. I put this slide here. It's an orphan slide, but we'll come back to it. Another really interesting thing that came out of this evolution experiment is that initially, when we put these populations in these 96 different environments, the phenotypes were all over the place, right? We had so much phenotypic variation at the beginning of the experiment. However, by the end of the experiment, regardless of environment, most of the organisms, most of the populations had converged on a single phenotype. Not a single growth rate, but a single phenotype. We measured things like cell size, chlorophyll levels and fluorescence, oxygen, evolution, C to N ratios. So these are all pretty consistent by the end of 350 generations of evolution, regardless of the environment, even though initially there was a really strong response to the change in the environment. So we saw widespread phenotypic reversion after 350 generations, despite the fact that we saw a lot of evolution, especially in these high NED environments here. So getting back to this idea of how right do we have to be with our scenarios, or are these high NED populations locally adapted? We did a crazy transplant. We took all of the populations evolved in single driver environments, and we put them in all the other single driver environments, and also all of the 5 driver environments. We took all of the populations evolved in 5 driver environments, we put them in all of the other 5 driver environments, and all of the single driver environments. And we got nothing. So what I'm showing you here, these are the blue bars are populations evolved in 5 driver environments. The red bars are populations evolved in single driver environments. This is essentially a growth rate. And you can see in the control environment, they have the same growth rate. In all of the single driver environments, they have the same growth rate. In all of the 5 driver environments, they have the same growth rate. And in the 8 driver environment, they have the same growth rate. This was terribly demoralizing because there was no sign that evolving with a single environmental change made you any more locally adapted than evolving to a case where there were 5 different changes, of which that change was 1, right? And we thought, well, this is terrible. Ecologists the world over are going to mock us. So we said, wait, we can look at local adaptation another way. And we said, we're going to look at how fast they grow at home versus how fast they grow away. So just home versus away. We weren't, so the laser is dangerously close to the forward button. So these are populations evolved in single driver environments, and if they're on the 0 line, that means they grow equally fast home and away. And you can see that in general, there's not a lot of evidence for local adaptation. These are the 5 populations evolved in a 5 driver environment, home versus away, and you can see that in general, we have 3 cases where it's statistically significant. They tend to grow faster at home, and they grow quite a lot faster at home. I would argue that this is local adaptation. In all of our experiments with clammy, the adaptive response to growing at high CO2 is to decrease growth. So if I had my way with the world, I would put a star here. Reviewer number 2 doesn't agree with me, so we're arguing about the star at the moment. Then we thought, okay, well, there's another way we can look at this. And this I thought was quite interesting. So what we did here was we had the populations rank the environments. So what I'm showing you here is each environment. The local population from that environment is in yellow. Foreign populations that come from a single driver environment are dark. Foreign populations that come from a 5 driver environment are light, and the control is black. I just want to point out to reviewer number 2, if you're in the audience, here's my CO2 single driver, and here's the local one. It's the slowest. It ranked at lowest. Oh no, it ranked at 1. There we go. And so I did actually reverse the CO2 one. Okay. It's all good. And so what you can see is there's no statistical difference in whether or not the local ones do better between the single driver environments and the multi-driver environments. There is, however, a big difference in how populations evolved in multi-driver environments do abroad in these single driver environments, which is that if you take populations from a multi-driver environment and put them into another multi-driver environment, they've got sort of even odds against everyone else. They're not particularly great, but they're not particularly bad. Whereas if you put them in these single driver environments, they are actually particularly bad. So it does look like they're locally adapted. Not in a way that I find super convincing, but there does seem to be some degree of local adaptation, and it's strongest in these populations that have evolved in the multi-driver environments, which means that they're seeing each 5-driver environment as a completely separate environment, more so than the single driver environments, which means that well, if this is any indication, it means that getting scenarios right is actually fairly important. I don't know how representative CLAMI is of organismal responses, but we did have enough environments here and enough different environments and enough different degrees of overlap between environments that we can attribute the variance to each of those and still see a local adaptation effect in the 5-driver environments. So just to recap, this is Fisher. Adaptive walks are Fisher's problem. And I like this picture of him because he thought that people with cancer smoked to alleviate the pain. So he got the correlation wrong. He wrote an entire treatise on the benefits of smoking. So he was really right about adaptive walks, really wrong about tobacco. He also thought women couldn't do science. So I enjoy giving talks with Fisher in it. So to recap, it's not harder to evolve with multiple drivers. It, in fact, looks a little bit easier. Populations from multiple driver environments are slightly more specialized. We're not sure why. So now I'm going to move on and talk about this thing that irked me, which is that the populations from the improved environments, so the high CO2 environments, for example, or for Chlamydomonas, the environment where we changed the light was an improved environment. Why do those never get better? Why can they never outdo the plastic response? They have mutations, we know this, right? And the second thing that irked me was that we saw trait reversion. All of these awesome phenotypic changes we documented at the beginning of the experiment with crazy high photosynthesis rates and changes in chlorophyll content, cell sizes, and changes in C to N ratios all just went away. So why? I only have a 20-minute attention span, so this is our break. All right. Break over. Is everyone refreshed? Does anyone else need to dance? No. No one else ever dances. So now I'm going to come back to this idea that evolutionary biologists, and I would argue, I've seen some ecology talks that make me think ecologists might kind of be the same way, like to think mainly about environments getting worse, organisms starving, or being killed by toxins, or edged out by competitors, or other sort of death and destruction, horsemen of the apocalypse stories. Which is all well and fine. I enjoy those as much as anyone else, right? And that's what our whole body of theory is there to deal with in evolutionary biology. However, environments don't always get worse. Often environments get better. Here I am experiencing an environment that improved drastically one morning. And I put this picture here because I wanted to remind myself to tell you yet another thing, which is that often when organisms experience environmental enrichment, they don't experience it from a point of view where they were like starving and nearly dead beforehand. They're sitting in an environment to which they're actually pretty well adapted, right? We know that because they're not extinct and no one has managed to outcompete them. So in a lot of cases, you have an environmental change where you have a population sitting in an environment that's actually pretty good. The population is actually probably pretty well adapted, right? It may not be the best environment in all realities, but it is the best environment available right there for that organism, probably, right? And then it gets better, right? And we're like, oh my God, I can't think of that as too awesome. So we generally don't. I blame Van Valen. You can blame your favorite evolutionary ecologist. But there's this, I'm not even citing this paper because if people don't know the paper, then I encourage you to Google, those of you with smartphones, immediately. But the idea is that environments are constantly deteriorating over time. And this is true if you want to make some interesting approximations and do some interesting theory. But what that means is if we look at a fitness landscape, so these are, you have traits or gene sequences, and the height of the mountain indicates how fit you are, we tend to think of populations as starting off in fitness valleys, when in reality, they probably start off fairly close to fitness peaks, even if there's an environmental change. This is because environmental changes tend to be, first of all, not novel, second of all, correlated in time. So the best phenotype tomorrow is likely to be quite close to the best phenotype today in many cases. Not always, I know flash floods happen. So we tend to think of evolution as going like this. You start from a point of extremely low fitness, and then in an environment that completely is terrible, you recover fitness and you go back to some growth rate that your ancestor would have had in this mythical, wonderful environment that they experienced. Does that kind of make sense? I'm exaggerating a little bit, but only a little bit. However, we see that often organisms can respond to environmental change positively. And what I'm showing you here is a great data set that Jeff Morris had a hand in putting together. And this is looking at responses of primary producers in the ocean in response to changes in CO2. So these are growth rate responses to ocean acidification. We have coccolithophores, not much of a response. Diatoms, not much of a response. Other large, which I'm assuming are these osteocracans. But what we see is that diazotrophs and Synechococcus have huge positive growth rate responses to CO2 addition. And then we take these responses and we say, okay, well, here's what we expect to happen over time. We expect that things with positive growth rate responses to take over. We have a world of Synechococcus, which will look like these green dots. I thought that was interesting. The other interesting thing about this data set is it incorporates both short-term and evolutionary responses to CO2 enrichment. And the evolutionary responses fall within the range of the short-term responses. So it comes back to this idea of why aren't they doing better? Why can't you evolve to do better than the plastic response when the environment is so clearly awesome and more awesome than it was yesterday? All right, so I did what every self-respecting evolutionary biologist did, and I came up with a hypothesis and gave it a funny name. I called it the prodigal son hypothesis, because I feel like we've gotten all we can out of Alice in Wonderland. So this is, it just keeps on doing that. This is cell division rate, or population growth rate. So here we have a population growing, and this is very important, in an environment to which it is well adapted, and not a bad environment. It's an environment where it's happy. The environment then gets better. High CO2, high chocolate, something, right? And then over evolutionary time, hundreds or thousands of generations, one of three things can happen. You can take this sort of plastic response and more or less fix it evolutionarily, so you can maintain the plastic response. This is what we see in long-term, in terms of growth, this is what we see in long-term experiments with things like chlamydomonas or trichodesmium. They have an initial positive response to high CO2, but they can never do better. They can maintain it, and in fact, that growth rate becomes irreversible. If you put them back in the old environment, they don't go back to the old phenotype. You can also reverse the growth rate response, and this is what I've seen a lot with pico-eukaryotes. I have yet to see an example of something that increased the growth rate over the plastic response over time. The reason I call this a prodigal son hypothesis, as you're going to see, is because the mechanism is that growing fast here, so living fast and hard, is actually in itself going to damage you enough to exert selective pressure to return to your ancestral, calm, sedate way of life, much like the prodigal son. So here's an experiment that where I'm going to just walk you through, because it's where we saw these prodigal son dynamics most clearly. And the reason we saw it clearly is that we used Ostriococcus tori. I think that's what we saw. And Ostriococcus is interesting. It's the world's smallest free-living eukaryote. It's only 1 micron. And more importantly, it doesn't have any spare parts. So it's got 1 mitochondria. It doesn't have a whole lot of extra space for chlorophyll. It doesn't even have a proper cell wall or membrane, right? It has sort of these scabby bits floating around on the surface. So it's not got a lot of wiggle room if things go wrong. So you can see effects of mutations really well. And what Elisa Shum did during her PhD with me is we evolved Ostriococcus for 400 generations in 4 different environments. One was high CO2 that was steady. One was high CO2 that was fluctuating. That was because we wanted to maintain plasticity. Then we had a control level of CO2 and control fluctuating. You don't need to worry too much about these, the fluctuations or lack of fluctuations. It's just that there's a high CO2 treatment and a low CO2 treatment. After 400 generations, we saw something shocking. So these blue ones here are populations that were evolved in normal amounts of CO2 in air. And here's how fast they grow at high CO2. So the plastic response is to increase growth by about 40%. And Jeff and I still haven't figured out why Austriococcus loves CO2 so much that it has this insanely huge plastic response. Yet it does. And if you then take those populations and evolve them for 400 generations at high CO2, you see that they completely reverse this growth response. They do it more if you fluctuate the high CO2, but this is at steady high CO2. So you can see that actually what they do is they completely reverse this initial party. And this is shocking for two reasons. One, this was a selection experiment done in batch culture, so we kept these things in exponential growth. This is what you do if you want to select for rapid growth. And what we saw was the deterministic reversion of this rapid growth response, which is pretty amazing. But it wasn't just growth. We noticed that a lot of other traits that had an initial response to CO2 reverse that response over 400 generations. So I'm just showing you, this is a bunch of traits. You don't have to worry too much about them. The important thing here is that if you compare this bar to these bars in the blue, that's the plastic response to changes in CO2. So you can see a lot of traits respond to changes in CO2. Some don't, but most do. And these traits are things like oxygen evolution, oxygen consumption, C to N ratios, chlorophyll content, blah, blah, blah. What the traits are is not super important. What's important is that after 400 generations, they've reversed that plastic response. So what they've done is they've reestablished the ancestral phenotype in the ancestral environment in the new environment. They're like, no, no, my phenotype was good as it was. Thank you very much. I've put it back. And I thought, okay, well, this happens repeatably and it happens rapidly, so it has to be adaptive. And what we did, in fact, find was populations with lower growth rates were better competitors. So what I'm showing you here, what I'm showing you here is growth rate alone, and here's the competitive ability of that same strain. So these are just different selection regimes. But what you can see is if the growth rate is low, the competitive ability is high. So there's a negative slope. In this case, we have a positive slope, but these growth rates are so high that they're just overgrowing everything we could throw at them. But in 3 of 4 cases, we actually see a really nice relationship where if you grow a little bit slower, you're actually a much better competitor, which is interesting. And I saw this in Chlamydomonas as well a few years ago and assumed that I was crazy. The other thing we noticed was that fast-growing cells are more stressed. This is a measure of mitochondrial potential. This is where the single mitochondria thing becomes important. The important thing to know is that if you're above the line, you're happy. If you're below the line, you're stressed. Here are the fast-growing cells, here are the slow-growing cells, and here are the control cells. So fast-growing cells are unable to maintain mitochondrial potential. When you have but a single mitochondrion, this is a big deal. This is the ability of Austriococcus to grow following heat shock. So what I'm showing you is the controls. This is their growth rate before heat shock. This is their growth rate after heat shock. So they don't like it, but they tend to survive. These are the growth rates of the controls in high CO2. So over very short time scales, CO2 is protective for Austriococcus. What you can see here is that the slow growers look kind of like the controls. They don't like heat shock, but they do okay. The fast growers have very low growth rates over heat shock, and I'm not showing you the data on this today, but the fast growers also only have about a 10% survival rate of heat shock. So these are the ones that make it. Most of them don't. So the fast-growing cells are poor competitors. They're stressed and they're wimpy. Excellent. I thought that good. So I tried to pull this all together, assuming that it was not likely that everyone in my lab was making the same mistakes on the same measurements all the time for like a decade. And I think that's okay, because, you know. And I also started seeing in other people's data sets this idea that you just can't do better than the plastic response. So I've seen phenotypic reversion in all of my high CO2 experiments for 16 years. So not all of these experiments have been set up to detect it, but if you go back and look at the data, it's always there. It's really surprising. Of course, there's some ascertainment bias there. The growth outcomes, however, are really highly repeatable. So when I see these responses, they're highly repeatable in terms of growth, but the phenotypic way that the populations get to that growth rate is highly variable, even within experiments. So high CO2 cells are often wimpy, if only for a little while. This is also true of chlamydomonas. When we went back and started staining chlamydomonas mitochondria, we discovered they were carrying a lot of dead mitochondria. Wimpiness can be overcome. Give it another 600 generations, it goes away. Growth rates don't go up, though. And I thought maybe there's a general explanation. Okay, so let's think about these short-term winners, short-term cases of happening upon a high chocolate environment. The short-term winners have a positive growth response to environmental change, which means they probably have to increase their metabolic rate, right? Which means they probably have to deal with more damage, more oxidative damage, or less ability to repair a normal amount of damage because they're dividing faster. This is just an energy allocation problem. And this damage could build up gradually if they can't step up repair rates. That's a lot of ifs, but there's also, I'm not the first person to come up with this. This is the entire basis of understanding the evolution of asymmetric cell division, also understanding the evolution of soma and germlines, right? So this is not my idea. And here I can show you, look at that totally cromulent paper, an aging cell, showing you have a cell, it has some amount of damage, it grows, incurs more damage, and divides. And it divides asymmetrically so that one offspring is going to get more damage than the other offspring. You can do this with repair. Don't memorize this model. I just want to show you that this is an old idea. It's an idea, the accumulation or avoiding damage accumulation underlies a lot of the explanations of macroevolutionary transitions in unicellular things. Most importantly, asymmetric cell division. And the idea is just if you can make a garbage can, you can make a cell that gets most of the damage, then overall you're going to be more fit because the offspring that gets the nice, clean, new, shiny parts is going to be in really good shape and have very high fitness. So I thought, okay, maybe that's what's happening here. Maybe what happens is that when the environment changes, you don't look at all of the traits as changing individually, but you think this organism was an exquisitely integrated network of trait values that work really well together. This organism was more or less optimized. They happen upon their pile of chocolate, or CO2, and they change not just their growth rate, but most of their other important traits, right? So suddenly this beautifully integrated system, it's thrown all out of whack, right? Now, you sort of from here have three options. These options are best illustrated with rock stars. So you change all the traits. You have the Keith Richards approach, which is that you can continually evolve ways to deal with living fast. And in fact, live faster and faster as you age, right? As you continue to evolve, right? So it means all the traits are changing, and what you do is you find a way to deal with those trait changes and change a lot of the way your traits are co-regulated, for example. Then you have the David Bowie approach, right? Which is an awful lot of evolution takes place, but you remain at the same steady level of awesome, right? So here you have a lot of evolution, but what you end up doing is fixing a lot of those traits. So you just maintain the plastic response, and so there's a lot of evolution going on, but it's mainly at the level of trait correlations and regulations, and you're just sort of turning this new normal into your exquisitely integrated organism. Alternately, we have the Paul McCartney approach, which is, oh my God, I can't do it, right? All of the traits changed, you're growing really fast, you're partying hard, and one day you just wake up on the floor and you're like, no more, I want to bed, right? And you just return to what you were doing earlier in life before the partying started and just reverse all the changes. And this is sort of the prodigal son. This is the prodigal son dynamic in the hypothesis. So I did what anyone would do before starting an experiment, which is I made a model. So I made a model where, just to see if the avoiding damage was sufficient to explain the dynamics I was seeing in the experiment. I'm not asking whether other things could or could not be going on. I'm just asking whether damage can drive this. And so I had populations. They had some damage. They would reproduce. They'd inherit their parents' damage. And they'd inherit their parents' growth. They'd inherit their parents' growth rate with some mutation. They would then grow, and they'd incur some damage as they grew. They would then reproduce. And every time they'd reproduce, they'd go through some sort of viability selection, which meant the more damaged you were, the less likely your offspring were to be viable. But they would also get more. They would also go through growth rate selection, which means the faster you grow, the more offspring you produce. So you can see that these two things are at odds. And what I did was I played around a little bit with looking at what happens if you allow damage and repair strategies to evolve or not. if we can change how viability works. And finally, I actually looked at how much of your parents' damage you inherit versus how much of that damage is due to your own growth rate. I'm just going to show you a little bit of the outcome, and the graphs are quite simple. So this is the growth rate. The old environment to which they were initially well adapted and which was fine allowed a growth rate of this, 0.8. I then changed the environment, so the new maximum growth rate that the environment could support was 2. What I'm showing you here, this log delta t, is essentially a degree of Lamarckism in the system. So if delta t is very small, it means that you basically inherit damage from your parents, and how fast you grow doesn't matter. A really low delta t up here means that you inherit damage from your parents, but it's immediately erased by your own growth rate. Somewhere in the middle means that you're going to inherit some damage to your parents and then mitigate that with your own growth rate, increase or decrease it, right? The color of the circle is just telling me the steepness of the relationship between damage and mortality. It's how likely damage is to, basically whether damage is likely to not kill you and then kill you, or whether there's a nice gentle slope there. And finally, the size of the circle, and this is important, is the chance that the population exists at all. So it's the chance that the population has gone extinct trying to evolve. And this is a reference scenario, and the reference scenario means that I've taken that population and forced it to live in the new environment where it can have a high growth rate, but I forced it to do it with its old damage and repair strategies and mortality strategies. So it's basically yesterday's organism in today's world. And what you can see is even though the environment has become enriched, the populations if their own growth determines the amount of damage they incur, they actually can't increase their growth rate very much at all in response to the environment increasing. They do transiently increase it, but this is after a few thousand generations of the model running. So here's the reference scenario. What you can see is that you can sort of alleviate this if you allow the populations to become damage tolerant. So here we go. These ones can grow fast now, whereas they couldn't here. You can make repair cheap, which I think might be pretty reasonable given that they now have more resources. So if you let them take those resources and you make repair proportionally cheaper, they can grow faster. You can also make them damage tolerant, and that works pretty well. And that could be the case if the new environment is somehow more permissive. So I think what this model showed in a very simple way is that we don't need to invoke any special voodoo for looking at the phenotypic or for explaining the phenotypic reversions we've seen. We can actually just explain it by growing fast itself being difficult. So I call this prodigal son dynamics, and just in case you wanted to, again, look at these beautiful pictures. I put this up just to remind me. There we go. And of course, what we see in individual cases isn't that the organism follows the same trajectory for every single trait it has. What we see is, for instance, with Austriococcus, many traits revert, but some don't, and some actually do go up over time. So it's a complex result. So I'm going to wrap up there, which is good because I have 35 seconds left. So stuff to keep me busy for the rest of my career, because I'm only about halfway through. So given population persistence, if it doesn't kill you, it's not harder to adapt to many environmental drivers. I'm not clear on why yet, but I'd like to know. The extent of this adaptation is really best explained by the initial drop in fitness, not by the complexity of the problem. So the other thing that I find really interesting is that this trait reversion is widespread across experiments. It's really interesting to me that natural selection that we would initially move a huge suite of traits, and then natural selection would essentially spend hundreds of generations moving most of them back. Like a child, like getting that one pea out of their pile of food, right? These prodigal son dynamics are consistent with being damage driven. I'm sure there's other stuff going on. This is my lab. These are the beautiful people who do the work. This is our website. And just in case anyone is interested, I also work with artists. So here's the Proceedings B paper that a lot of the Austriococcus data is in. This is a manuscript in a way that you usually see it. This is a manuscript, sort of an older way of writing that same manuscript. And this is a collaboration with some artists, and we're hoping that this will go into the National Gallery of Scotland in some sort of art science thing. There's also, interestingly enough, music being made out of the data to go along with this manuscript. I'm not sure how that sounds yet. Thanks.
Thank you very much. So if you have questions, would you please come up to the microphone so we can hear your questions, or wave and I can come running?
Rebuttals are also welcome.
Hi, great talk. I was wondering how you think this sort of framework fits in with a community ecology perspective. So in environmental communities, you can see high rates of species turnover, obviously from selection and drift. So do you think that would facilitate evolutionary processes, or in some way trump them, so this it has less of an effect in a super diverse community?
So that's adding a level of complexity. Well, so we started looking at that by working in mesocosms.
Okay.
And the effect of having other species present does change the evolutionary responses of focal species when we look at them. And certainly there's a lot of data now saying that a lot of the control, especially microalgae, is top-down from viruses and grazers and things. So it will add a level of complexity. So you can be really flippant, and you can say, well, it's just another driver. But it's unlike environmental drivers, these evolve back at you. So I imagine it's going to make it even more intractable. But I am game, we're trying it.
Cool.
So I don't know is the answer.
Many organisms have never been cultured in the lab.
Mm-hmm.
And I have wondered whether those organisms that do lend themselves to culture in the lab have certain properties in common. Like you can ignore them for a couple of months and they'll still survive, and you can reestablish their populations. And so I'm just wondering whether you have any thoughts about whether perhaps we have done some sort of selection just by which organisms are common laboratory weeds.
Right. So I think a lot about this. And I think we've learned a lot from model organisms. I think the trick is to extrapolate wisely from our experiments. I would not ever say, and would never support someone saying, that the actual phenotypic changes I see in Chlamydomonas predict the phenotypic responses of natural populations. This is a petri dish in an incubator for something that has been really strongly selected for growing in culture. However, the laws of evolution are not different in the lab and the field. So I think the trick is to design your experiments intelligently, and then to understand which bits of your experiments are generalizable and which bits of your experiments are specific. And that's the reason I don't go into the specific phenotypic changes in these Chlamydomonas culture lines. It's not important and it's not relevant. What is important and relevant is the fact that they can't seem to, is that to them, when you take these 5 driver environments, they see them all as completely different. That, I think, is probably not a lab artifact. Or you get this idea where we see widespread phenotypic reversion across experiments. This we've seen in Austriococcus, and we've also seen it in culture in diatoms that we've taken freshly out of fjords. And so that's been consistent in Chlamydomonas, Austriococcus, freshly cultured diatoms, and now in a bunch of communities that we've isolated where we don't even know where most of the species are. They're just O2s, right? And so I do think that these model systems can tell us a lot. You just have to be super careful about how you extrapolate. If someone told me, aha, this means photosynthesis rates will go up by 20%, I would just laugh at them. Photosynthesis rates will go up 20% in a petri dish in an incubator with this genotype. How's that for now? Does that get you? Yeah.
Other questions? I have one.
Excellent.
Oh, where? Okay. So we typically use growth rate as a measure of fitness when we work with microalgae. Should we use something else? And if so, what would we use?
This is the million-dollar question. I mean, so you can use competitive ability. That's not always logistically possible. You need a marker. I think what you have to do is you have to use multiple measures of fitness and try to figure out what you've actually selected on in that experiment. So I think one of the reasons doing work with E. coli is so incredibly popular, or working with initially degraded environments, is that you know selection is for increasing growth rate, because you're essentially reestablishing a growth rate. You're not going into a completely new one. So unfortunately, I think you actually need to think about the biology of the system and the actual experiment you're doing and try to figure out what you're selecting. And the best strategy that I've been able to come up with is using multiple measures of fitness and then putting together a story of what natural selection is actually seeing. Sometimes it sees a slope, sometimes it sees the shape of the growth curve, sometimes it sees competitive ability. It's also a question of what you can do. But one thing that I think is worth noting is that growth rates are used as a measure of fitness, so that tells us about an amount of adaptation, but you can actually get an idea of how much evolution has happened just by checking whether or not changes are heritable, right? So it also sort of depends what your question is. Whether or not that change is adaptive may not be your main question.
Hi, really wonderful talk. I got a little hung up in the first 5 minutes of your talk because you posed the question of the number of environmental drivers and the rate of adaptation. Mm-hmm. And if we go back to Fisher, rate of adaptation is the product of heritability and selection differential, right? So, so your punchline at the end was, yes, the strength of selection. So if you are testing this hypothesis of the number of environmental drivers, are you inherently assuming that all of them have the same strength of selection?
No, we're assuming that the strength of selection from a given driver is independent of how many other drivers are present. So, and that's actually based on some short-term work we did, which is that the interactions between drivers are present, but for large numbers of drivers, they sum to approximately 0. So that's the assumption. I think the main problem with using just straight up undiluted Fisher to look at problems like this is it makes a lot of assumptions about pervasive epistasis, which we know aren't true. And the second assumption it makes is that when you have more traits under selection, mutations have smaller effect sizes on individual traits. And I see absolutely no biology supporting that. So it sort of assumes that if more traits are under selection, that individual mutations are going to have smaller net effects on fitness. And if you sort of back out of that and look at what that means, it means that if you have a trait that affects color and you're selecting out other independent traits, a color mutation would have less of an effect on color. So for independent traits, the assumptions about the way the magnitudes of mutations scale doesn't really hold either. So I think the reason we started with the whole just like undiluted Fisher framework is that it gave us a really clear way to design the experiment. And we sort of had the framework has known problems and that we can go in and check.
Right.
If that makes any sense.
But your punchline, again, in the end was it's the strength of selection that matters.
Right. So the strength of in that case, I'm not quite sure I understand what you're getting at.
Well, so if I have X number of drivers, right, but one is a hammer and the other ones are very subtle, it doesn't really matter how many subtle touches you give relative to the hammer.
So here's the question. So that's actually the short-term paper that's already published. It's been published for 3 or 4 years at this point. And we did find, in fact, that the short-term response was driven by the single dominant driver in a system, but that was only true when there were large numbers of drivers. So if there's 2 or 3 drivers, the interactions between those drivers are incredibly important, and you get a lot of non-additive effects. If there are 7 drivers, all you need to know is the worst one. And it's not that the other drivers don't have any interactions, it's that those interactions sum to 0. So what you're saying phenomenologically holds up, but the mechanism we uncovered isn't consistent with that.
Thanks.
Okay, so if we don't have any more questions, we'll break for the afternoon. We will reconvene tomorrow at 8:30, and we'll give Sinead another hand of applause. Thank you very much.